on your computer.
Why?
It doesn't work on here.
I bet we can get it to work.
No, like Mom.
Mm.
You don't have. It doesn't work. I tried every button.
Look.
What? Oh yeah,
Why is it jumping and why doesn't she have a
Mom.
head?
Oh, because it's headless.
I don't. What?
Mom, give me it.
Okay, now I'm not jumping.
Mom, give me it. Mom.
How do I walk?
No, Mom. Mom.
What, dude?
What
here.
did you do to get in the game? Here, let me try. Give me the remote. You just like how the male Mom, Mom, what did you press?
I don't remember. Hang on.
It's like a cow.
Okay, X makes you jump. This moves around your camera, but I don't know how you walk.
Mom,
Well,
see
I can't
this?
go anywhere. I did no. Look, that moves around the camera.
Then
That does not walk you.
if you can get out and then do it on your computer, and then you can just step on here. I guess.
Oh. Get out of here.
You're playing on the computer for a little bit.
I can't, dude, because I'm on here. Yeah. All right. All right. Hi, everybody. Dogs are going crazy. So let's go ahead and get started first with going over the I'm trying to get my stuff together. The commonly missed stuff, and then we'll come back to talking about the rest of it. Gosh, I'm sorry, my dogs are going crazy right now. It's the wind, man. They think the wind is something all the time, and it's not. It's literally just windy. Okay, so let's go over the commonly missed stuff for week 9, which is chapters 11 and 12, and we just went over these and kind of talked about the notes that I would have made and everything like that. So let's see. For chapter 11, we talked about microbial control and sterilization versus disinfection versus asepsis versus decontamination. And a brief rundown of that, of course, is going to be sterilization kills everything. Disinfection does not kill everything and is usually used on inanimate objects. Asepsis is the same thing as disinfection, but on living tissue. And decontamination is going to be mechanical, like actual physical removal of those microbes. We already know the difference between static and cidal, right? Static, no growing. Cidal, we're killing them. We have being aware of stopping how microbes can grow and stuff by inhibiting their protein functions. And we can do that by deactivating their proteins through denaturation. And denaturation is essentially the same thing for proteins as we learned about with DNA, just ruining the bonds that normally keep the shape, and the shape of those proteins is extremely important for their function. Thermal death time is the amount of time it takes to kill a microbe at a specific temperature. And thermal death point is the temperature that you need to kill a microbe if they're only exposed for 10 minutes. Then we have the different ways of removing the physical versus the chemical. And then some notes under that. So we have the physical and mechanical ones being like heat, and then that's going to be dry versus moist. We would also include radiation in that area, filtration. We didn't talk much about the ultrasonic waves, but that's a thing. And I'm not ever going to really ask you about it, so don't worry about it. But then cold doesn't kill anything. It's better for preservation, as is desiccation, but it can help preserve your foods. And then osmotic pressure using hypertonic environment, such as packing in salt or high sugar. Boiling doesn't kill endospores. It doesn't sterilize. So remember, if you want to sterilize, you have to kill everything, and that includes endospores. But if you can kill endospores, you're going to kill everything less resistant than that. So everything below it on the little chart that we had shown in that chapter. The chemical guys, we really talked about halogens, which are, we focus on the chlorines and the iodines, the phenols, the alcohols, hydrogen peroxide, the detergents and surfactants, and quaternary ammonia compounds that are going to get into the membranes and disrupt them. The heavy metals that are going to bind in the active sites of proteins to stop them. Aldehydes, gases. We didn't talk about dyes in the lecture, so don't worry about those, just like the ultrasonic waves. And then the acids and alkalis. Of those, if I were just to give a quick rundown, since there's not really a true list on here for that, for the chemicals, the ones that can potentially sterilize if you're using the right concentrations and using them properly for the amount of time that they need to be used, we have the halogens can sterilize, the hydrogen peroxide at high concentrations can, and glutaraldehyde and sometimes formaldehyde, depending on its use. The gases can sterilize, and that's it for the chemicals. The other ones can't. All right, when we're talking about physical methods, the most common ways to treat are going to be heat, that's dry or moist. We use autoclaves most commonly in medical situations to sterilize things. Those use moist heat and pressure in order to work. We also have dry heat, incineration, and incineration and dry ovens. They both sterilize along with that autoclave. But filtration can sterilize as well. It's a mechanical removal method, but it can sterilize if you have the pores in your filters are small enough to literally remove any living creature, then of course you can sterilize. Pasteurization cycles between temperatures until you have killed enough pathogens that it's not harmful anymore. We kind of already talked about the halogens. Phenol is pretty toxic and damaging. It was first used by Joseph Lister to treat, operating rooms to reduce. Hi, what are you doing, Ripley? Weirdo. To reduce contamination there. No, don't do that. Don't get in my face right now. Then we have the alcohols that will disrupt the cell membranes. They also disrupt the cell wall. And at concentrations of 70%, when we have some of that water present, it can actually coagulate proteins as well. So they can be pretty effective, but they don't sterilize. And isopropyl is more effective than ethanol. The oxidizing agent that we talked about really was mostly just hydrogen peroxide. These guys work the best on anaerobes because they're not equipped to handle oxygen. They wouldn't need to be because they're anaerobic. And most of the anaerobes can't even survive in the presence of oxygen at all. Cool. Then we have, and you can actually use hydrogen peroxide at high concentrations, vaporize it to sterilize things, almost like the chemiclave that we learned about with the ethylene oxide, the explosive gas. We usually don't use it in large rooms quite like that. We'll use it more in like they say, sterilizing cabinets. So we have surfactants, and not everything can withstand that oxidizer. So it can be damaging to some kinds of metals and stuff. Surfactants are just sudsy type of things. Okay, so if it sudzes, it probably is going to be a surfactant or detergent. Detergents are surfactants. And those guys have a charged head and a nonpolar tail, just like your phospholipids. And because of that, they can mimic a phospholipid, insert themselves between your phospholipids and your lipid bilayer, and therefore weaken your membranes and cause leaking of the cell. And so it's effective that way, but they don't sterilize. However, if they can cause leaking of the cell, they can also allow things into the cell. So they usually come in combination with other things. And most of them work better if they have a positive head, so that they're even more attracted within those negative heads of your phospholipids. And those ones that, with the positive head tend to be the quaternary ammonia compounds, the quats. Let's see, aldehydes, they're going to affect the enzyme function. This includes glutaraldehyde, which can absolutely sterilize. Formaldehyde, or the aqueous form of it, we have formalin. So it's toxic and we use it for embalming, but it's not as good at sterilizing. You have to use it in more dangerous concentrations if you were going to do that. So we don't typically use it for that. And then orthophthalhaldehyde, which is going to be less irritating than glutaraldehyde. It's faster, but it's more expensive and it's not as effective. It's not sterilizing. Ethylene oxide as a gas we can use as a chemiclave, filling a whole entire room full of stuff, filling it with that gas and sterilizing everything in that room all at once, but it's explosive and carcinogenic. We had also chlorine dioxide, which had a similar effect, but we used this in way more applications like treating water and things. Then we had acids and alkalis that are going to disrupt your pH. If you remember about how proteins work and rely on hydrogen bonds, then you know that those are going to affect the ability of those and the ability and the stability of those bonds to work in favor of those proteins and maintaining the structure, which is important for their function, right? Okay, so I'm just going to check on, see if we have anybody in the chat. Brittany's face is going to pop up while we're doing that. I don't know why I can't just always just have it like attached. I'm assuming you guys would say something to me by now if you couldn't hear me. So it's hard for me to know. Sometimes it doesn't give me the little icon on this. Anyways. All right. Chapter 12. Look at this nice little list. This is why I like this one better. So there were a lot of slides in chapter 11. A lot of it's just kind of memorizing little tiny groups of stuff. Chapter 12, we kind of have specific antibiotics and drugs that we just need to be aware of how they work. So that makes it a little easier. The concepts we had to know about selective toxicity, like we had said in class, being selectively toxic to the microbe and not toxic to the patient. And we have reminding you guys that antibiotics treat bacterial infections and not any other kind of microbe. Penicillins and other beta-lactams, like the cephalosporins and carbapenems, are going to affect the cell wall. This is common drugs to know, but yes. So I don't know why this isn't down below, but hey, this goes below that. Penicillins and other beta-lactams. So anything ending in cillin, or I will straight up call it a cephalosporin or a carbapenem. Okay, so it's pretty straightforward. Affect the cell wall synthesis. The ones that affect ribosomes are going to be the aminoglycosides and the macrolides. These are the protein synthesis inhibitors. Tetracycline is a type of aminoglycoside. And then we have the mycins, most of which fall into macrolides. To inhibit folic acid synthesis, which our cells don't do and the bacteria do, it's a good target, just like the cell wall. We would reach for sulfas, and trimethoprim is going to fall under this category of the folic acid inhibitors as well. We have the drug one that we needed to know that affects the cell membrane, and that is polymyxin. It's kind of toxic though, especially to the kidney. The ones that inhibit, because obviously we have cell membranes, and it's kind of ubiquitous as far as cells go. Same thing with the nucleic acid. So if we're inhibiting nucleic acid synthesis by blocking the helicase enzyme that unwinds your helix, so we can split it open and go through replication and transcription, then that could affect your cells. The guys that do that are called fluoroquinolones. That's the group. And then the drugs that fall into the fluoroquinolone category that you need to know end in floxacin. So that includes ciprofloxacin and levofloxacin and similar drugs. That was it for antibiotics. The antivirals, we talked about Tamiflu, and we talked about Rolenza, which is basically Tamiflu, just FYI. They just block neuraminidase function of influenza, which for our purposes, it's how the virus gets into cells and how it buds off as well. So if you can block that, And like we had mentioned in my late class last night, that's something you want to do early in infection, because once it gets into the cell, it's going to make its products in there. So the sooner you can block it from getting into cells as much as possible, the better. That's why you want to take it early in the infection. Otherwise, it's just already at the point where it's not going to help a lot. Acyclovir is an antiviral that blocks herpes. So HSV is herpes simplex virus. And so it'll block the nucleic acid synthesis in that. And then we have HIV antivirals that are going to target the aspects of HIV that are unique. And there's 3 traits about HIV that are unique. That is going to be the reverse transcriptase, going back from its RNA to the DNA that inserts into your DNA. If it wants to insert into your DNA, it also needs an enzyme called integrase that integrates it into your DNA. And it also has another protein called protease. It makes its basically its protein translation product in one big old long amino acid sequence, and then just cuts it into the pieces that it needs. And that thing that cuts is the protease. We don't need any of that in our cells. So if we could target that, it'll be effective at stopping HIV from making new virus particles. But once it's in your cells, there's really nothing we can do for it. So we can't cure it. All right, antifungals, the ones you need to know are amphotericin B, and then the azoles in general. And remember, there's one azole that we learn about in the chapter that is not an antifungal, but is an antiprotozoa, as well as being used for anaerobic bacteria infections. And that was metronidazole. Metronidazole, they went to the big city, which is moving up in the world to the protozoa. And having more functions as well, protozoa and anaerobic bacteria, very useful. So it's on its like own little special category there. But then we have the other antiprotozoa category for malaria. And those are quinines and quinine-based derivatives that end in quin. Like chloroquine is the one that I will probably ask about on the exam if I name one specifically, or if you get the question that names one specifically. The helminth drugs, the ones that I would have you guys remember, because it's the ones that come up the most, usually in the veterinary field, are going to be praziquantel and ivermectin. Then we have using Kirby-Bauer assay, finding the zone of inhibition around an antibiotic to determine the minimum amount of that antibiotic to inhibit growth, which is called the minimum inhibitory concentration. Then the therapeutic index is the ratio between the dose of the drug where you start having toxic effects versus the dose of the drug that works. So you don't want them to be too close to one another, because if you need this much to make it work, but that's very close to how much you're going to get to start having toxic effects, you might want to pick a drug that they're not so close together. Yeah, when you talk about side effects, could be, damaging. It could have allergies, things like this. Super infection is when you kill off the normal bacteria in that area, and then what's left will overgrow. So your good bacteria taking up the stadium seats, you take your antibiotic, you clear out those stadium seats. Now yeast can overgrow, or really resistant microbes like Clostridium difficile, C. diff, they can overgrow. So that's super infection. We have drug resistance that can be intrinsic. You have to be resistant to the antibiotic that you make. If you are producing penicillin to keep microbes away, then obviously you can't be sensitive to penicillin. That's just ridiculousness. And then acquired drug resistance, which is what we normally think of. You got it from somebody else giving you the genes for it, which you would get via conjugation through the sex pili, the channel between some cells, and shuttle it between. Then we have transformation, which we already were, tested on in the lab. That's, if you guys didn't see the announcement that I posted, good job on that average. I mean, that is not an exaggeration when I tell you that you have increased the average by 17% from the, even the summer, when our summer people do well. Even from that. So that's no small thing. And I know people say, oh, well, it's the study sheets and maybe that's all you studied and that's great, but still you did it. The other people didn't. So be proud of that. And if you still feel like you didn't do as good as you wanted, you probably still did better than you probably would have. if you hadn't done what you did. So now we know moving forward the type of, maybe notes that we need to be focusing on. We'll talk more about that after this. Anyway, so shotgun approach, overprescribing, livestock use all play a part in developing resistance. Oh, and we also forgot about transduction. I'm sorry. We know about transformation, picking up stuff on the outside, but transduction is that word that we use for when phages, bacteriophages, which are viruses, come and infect bacteria that they target, insert their DNA, the prophage, into those bacteria, and then undergo lysogeny, which is just hiding out like that. And they can carry genes into the bacteria and give them new traits such as antibiotic resistance. That term for that is lysogenic conversion. We learned it way back in like chapter 4 or something, chapter whatever it is, chapter 7, viruses, that one. All right. We did this. Okay. And then, yes, if you were going to be resistant, mechanisms that you can act by include inactivating the drug, preventing uptake, preventing the ability of it to be able to get into the cells, eliminating it immediately if it does get into the cell, reducing its ability to bind to its receptors to get in, or changing the metabolic pathway of usually folic acid synthesis to avoid the drug. Okay, this one's not as long as some of the other ones. Chapter 11, the most missed concept. One of them is going to be methods that sterilize. Make a flashcard. Make a flashcard that lists all of these things. Autoclaves, moist heat. Yes, that specific version of moist heat treatment. Dry oven and incineration. Ionizing radiation is great because it penetrates. Non-ionizing radiation will work on the surfaces only. It doesn't penetrate. Ethylene oxide, explosive carcinogenic gas. Chlorine, iodine also falls into that, just saying. And then hydrogen peroxide, formaldehyde, and glutaraldehyde especially. And then filtration. Methods that don't sterilize is anything else. And then I had our little mnemonic, which was I dare chaff, which is just going to be talking about, incineration, dry oven, autoclave, radiation, ethylene oxide, chlorine, and iodine, other halogens, hydrogen peroxide, aldehydes in general, and filtration. That's what that stands for. So our questions, which of the following methods is most appropriate for sterilizing? Pretty straight up question. And our choices are ionic radiation, boiling, rubbing alcohol, and pasteurization. We already know boiling doesn't kill endospores. Rubbing alcohol, it's effective at controlling the microbes, but it's not going to be sterilizing. And pasteurization is pretty terrible at even getting remotely close to that, but it does preserve the nature of liquids, taste, flavor, and what it does for you in your body and everything like that. When we talk about milk specifically, but it removes the pathogens, but it doesn't sterilize. We know ionic radiation, which is going to penetrate, and eventually, if you're exposed to it long enough, could potentially damage your DNA to the point of causing cancer. If you are going in for your cancer treatments and you're receiving radiation, it is going to be ionizing radiation because they're trying to focus that horrible ionizing X-ray energy at your cancer cells. If we can destroy the cancer cell DNA, then it can't be cancer. So it can go even beyond causing the cancer to then eventually destroying the cancer. So it's that, I don't want to say scary, but yeah. In a clinical setting, which sterilization method is commonly used to ensure surgical instruments are free of all microbes, including bacterial endospores? And we had said, obviously, the surgical setting, you're going to reach for autoclaving. UV radiation is great for just surfaces that the light is, aiming on, but it's not going to get underneath that item and it can't get through that item very well. It's just not going to be entirely useful for treating everything. The most effective thing will be that moist heat with pressure in that autoclaving situation. So it'll be this one. and it'll be this one. All right, next we have osmotic pressure. Osmotic pressure is going to be subjecting cells to environments where solute, like the stuff, right? Not the water, but the stuff that's in the water or other liquid, is higher outside the cell than inside. So packing in salt and sugar. We know you can pack your foods in salt and sugar to control microbes. So that's called osmotic pressure. Don't forget what osmosis means. Osmosis means movement of water across membranes, and we learned it in at the same time as we learned hypertonic and isotonic and hypotonic. So don't forget about that. That's all this is talking about, is the salt environment around cells or the sugar, but it doesn't matter. Whatever it is, if it's higher outside than inside, the cell's going to give up its water from inside to try to water down its environment on the outside, which seems ridiculous, but that's what chemistry and physics has it do. And so that's the idea of how that will work. So it's great for food preservation, but it does not sterilize. So our little mnemonic was supposed to be salty jam stops microbes. I mean, it speaks for itself. Salt, sugar, those things will stop microbial growth, but it won't sterilize. How does osmotic pressure help in controlling microbial growth and in food preservation? Does it alter the temperature? No, right? We're just going to knock that one right out. No, it does not. Okay. By increasing salt or sugar concentration, and that'll cause water to leave the cells. We read that whole option, and this one sounded pretty good. By introducing antibiotics, no, we're not introducing antibiotics with osmotic pressure. You think about osmosis, that doesn't involve antibiotics. Osmosis doesn't involve temperature. Or by exposing microorganisms to UV light. Last time I checked, osmosis doesn't involve UV light either. So one that does involve osmosis is B, because we have water leaving the cells. That's because of packing it in salt or sugar. That's B. Which of the following statements best describes why high osmotic pressure is effective? So does high osmotic pressure directly destroy DNA? It shouldn't. Does high osmotic pressure create an environment with low pH? It shouldn't be changing the pH. Not upfront. High osmotic pressure, does it cause microbes to lose water, then lead to dehydration and eventually cell death? This is even a process. It's called plasmolysis. We learned about it before in chapter 9. That's that shrinking and shriveling of that cell. Cool. That's going to be that one, obviously. But high osmotic pressure, does it enhance the growth of bacteria? This is nonsense. All right. So that's easy enough. Packing things in salt and osmosis. Those things should automatically in your mind be linking, because if you fill your mouth with salt, it's going to suck all that moisture out of your mouth. It's the same idea if you put just cells in there as well. And that's osmosis that's making that happen. All right, next let's talk about hydrogen peroxide. We said about how it is oxidative. We can use it as an antiseptic and a disinfectant. Remember, antiseptic on tissues, disinfectant on inanimate objects. You're going to usually use it at home at concentrations of 3%, and then industrial will use it for sterilization at about 35%. That's the one that's going to be actually truly sterilizing. So it's going to create reactive oxygen species. These are going to be free radicals, and that is damaging to cellular components. I don't know what to say, but it can lead to cell death if you can't manage those free radicals on your own. So our cells are equipped to deal with that because we have to be able to in the presence of oxygen all the time and using oxygen all the time. But anaerobes are not equipped for that. They don't usually need to be in their normal environment. So this can be very effective on anaerobic bacteria. It can be also effective against other organisms that don't have something in place to deal with a higher level effects of high concentrations of hydrogen peroxide as well. So You need higher concentrations to be sterilizing if you want to get rid of endospores, for example, because they're going to be pretty resistant. So yeah, let's see. You do need higher concentrations to sterilize. It can be corrosive, and potentially you can have some resistance developing. I mean, you don't see a whole lot of it occurring in microbes, but it can happen. So H2O's bold vows for endospores. Basically saying, one of the products we're going to be making from hydrogen peroxide, H2O2, would be water if we're able to deal with it. However, if you're not able to deal with it with your catalase enzyme, then you need high concentrations to be able to get rid of endospores, but you can still use it as a disinfectant. What's the primary mechanism by which hydrogen peroxide acts to control microbial growth? Does it disrupt microbial cell wall by enzymatic activity? Basically saying, does it act as an enzyme to disrupt the cell wall? None of the above. It doesn't. It's an oxidative thing. It's going to cause free radicals that cause cell damage. So it causes cell death through the production of reactive oxygen species. Of course, that's the one, but does it alter pH of the environment, inhibiting microbial enzymes? It shouldn't be really affecting it that way as much as it is doing these free radicals, okay? And then we have formation of a physical barrier. That's ridiculous. No, there's no physical barrier. And I'm not sure any of the treatments that we have create physical barriers. So it's going to be B. In a healthcare setting, how is hydrogen peroxide commonly used to ensure the safety of medical equipment? So now we're talking about using hydrogen peroxide in a medical setting. Do we use it to coat medical instruments to prevent microbial contamination during storage? Do we apply it directly to wounds to speed up healing by reducing microbial load? Is it employed to disinfect surfaces and sterilize equipment, especially through its oxidative properties? Or is it added to solutions to enhance the effectiveness of other disinfectants? So I think we rule out D for sure, right? Because the things that you're adding to solutions to enhance other disinfectants typically isn't going to be something like an oxidative drug or oxidative chemical, excuse me, because it's also going to potentially be oxidative to other chemicals. So we don't see that, and we haven't talked about it that way. So it's not going to be D. The ones above it, though, get tricky, and it's a matter of understanding how it works best. In a healthcare setting, are we usually coating medical instruments to prevent microbial contamination during storage? No. When you expose something to hydrogen peroxide, it works in that moment. It doesn't carry the effect with it afterwards. As soon as you're done treating it, you can go throw it in the dirt, and it's not going to be any more protected from microbes than anything else. So it's not going to be that. It doesn't have a lasting effect. Is it applied directly to wounds to speed up healing by reducing microbial load? Versus, is it employed to disinfect surfaces and sterilize equipment through its oxidative properties? Typically, we're going to be looking at C. And this is what, number one, it says oxidative. So we know that's kind of a giveaway. Sure, that's enough for you. That's great. But the reason it's not B is simply because we're only going to be using it as a antiseptic in the 3% concentration. And at that low concentration as an antiseptic, it's not going to be super dangerous to aerobic compounds. And if you have a wound, especially at the surface, those are either aerobes or facultative anaerobes. And facultative anaerobes are the ones that switch between the aerobic form versus fermentation. So they have to be also, equipped for dealing with oxygen. Those guys are not very well targeted by hydrogen peroxide. Only deep wound things are going to be. And even then, if you're wanting to be effective, you would need high concentrations, and those high concentrations are just not going to be effective. Are just not going to be safe for use on your own cells, because at some point, even your own cells can't handle, 35% concentration of hydrogen peroxide. It'd be ridiculous. So it's not going to be this one just because of how the application of it, it's not super effective that way. Can you use it to treat wounds? You could, but it's just not going to be very effective for it. All right, we have antimicrobial. It's going to be more commonly used, I should say, to sterilize equipment. You know, the ones that can't go in the autoclave necessarily might be able to be used in a sterilizing cabinet with that hydrogen peroxide. All right, next we have the drugs. So chapter 12, we have therapeutic index. Again, that is the concentration of drug that's going to be toxic to you versus the concentration of drug that's going to be effective when you take it. So if it takes 0.5 milligram tablets to treat your infection, minimum, but 0.5 milligram tablets are also more likely to be where the threshold is for causing toxic effects, that's a concern. So you want to be sure that you are, hitting that level that you need to be effective at a much lower concentration than where the toxic level is going to be. So that's what therapeutic index is talking about. The effective dose, where you're getting that desired effect, versus the toxic dose, where you're getting the harmful side effects. Yes. So then it talks in this like breakdown about the high versus the low therapeutic index. High therapeutic index means there's a larger margin of safety between the effective versus the toxic. That gives you a lot more wiggle room for safety. The low therapeutic index means that there is a very small margin of safety and it becomes more dangerous. So you definitely want to think about this whenever you're developing or choosing the drug of use. So again, the mnemonic here that I had was just TI. Sure, therapeutic index, but just remember that therapeutic index is your toxic effective ratio. So what does a high therapeutic index mean? Is that drug at a high risk of causing severe side effects, even at low doses? Is the drug having a large margin of safety between effective and toxic doses? Is the drug effective only at very high doses, making it less safe? Or is the drug's effectiveness closely tied to its potential for causing resistance? So if we read through these again, the first thing that we want to remember is that if we make our notes, we would write that therapeutic index relates to the dose that's effective versus the dose that causes bad effects. And the closer that those are, the more dangerous it is. So the farther away that they are, the safer it's going to be. So easy. So that's great. But which one's the high therapeutic index? Think of that as being the highest distance between those two things you're measuring, effectiveness versus toxic. And the farther apart they are, the higher that distance, the better. Okay. So that's going to bring us to large margin of safety. That's going to be B. Sorry. Oh, what? I don't know what that was about. Okay, that's going to be B. No. In a healthcare setting, why is it important to consider therapeutic index of a drug when prescribing medication? To determine the drug's color and taste preferences? Get out of here. To ensure that the drug is administered at a dose that maximizes the effects while minimizing toxicity. Yes, now we're talking. Do we want to, is therapeutic index important for evaluating cost effectiveness? Get out of here. Or to predict the drug's effectiveness. And if these are ridiculous choices. B, obviously. Okay, moving on. Targeting protein production. We have the groups of the aminoglycosides. You guys don't need to worry about streptomycin and gentamicin. I promise you that this isn't a concern that you need to worry about. The aminoglycoside related one that you need to know about is the tetracycline. Okay. So aminoglycosides, tetracyclines, and macrolides all work by, that's why I didn't bring up those other examples, because I didn't want you to be confused. I want you to worry about the mycins and the macrolides. Okay. And then the tetracyclines falling under that aminoglycoside kind of umbrella. Anyways, they all affect the ribosomes. So this is a lot way more complicated way of saying all that. So you have, come on, dude. I keep, I guess, hitting the wrong thing. So you have protein affecting guys. That's going to be aminoglycosides, umbrella, and then the macrolides. Macrolides are going to be the micins. Some of them, again, MICINs, but it doesn't matter. The only one, most of them are going to be falling into that category of micins underneath the macrolides. Those are the only ones I want you to worry about, please. For the aminoglycosides, ones related within that category anyways, are the tetracyclines. Okay, turns out all of them that are going to inhibit protein synthesis via stopping ribosomes. All of them work that way. All of them work that way by stopping how the ribosomes work. Now, if you look through here, even in this broken down, I broke it down in a lot more detail than what you guys even needed to worry about, but they all mention about ribosomes, and you don't need to know the exact mechanisms of it. This that I wrote out here at the left is all you need to remember. If you're going to inhibit proteins in, that's always the way that I say it sounds weird. If you're going to be inhibiting protein synthesis, you're going to stop ribosome function. Both aminoglycosides and macrolides work that way. The aminoglycoside related drug that you need to worry about is tetracycline, and the macrolide drugs that you need to worry about are the micins for our purposes. Which of the following antibiotics affects protein production? Carbapenem, aminoglycosides, Cipro, or metronidazole. We know carbapenem is going to be cell wall. Aminoglycosides is going to be the answer, protein via ribosomes. Ciprofloxacin and nucleic acids. And metronidazole, we usually learn about this as being protozoa and anaerobes, but it works through nucleic acid stuff. You don't need to know that though. But yeah, it's going to be aminoglycosides. Okay, why might tetracycline be preferred for treating gram-negative infections as opposed to amoxicillin? So amoxicillin is a psyllin, and psyllins affect the cell wall synthesis. So amoxicillin is easily metabolized by the gram-negatives, or tetracycline targets protein production while amoxicillin targets cell walls. And gram-negatives have very little cell wall. Amoxicillin enhances protein production. That's just ridiculousness. Or tetracycline attacks gram-negative outer membrane. That's also ridiculousness. Okay, so we know tetracycline deals with the protein production, and amoxicillin being easily metabolized by gram-negatives is not the concern for why it's not a good choice for gram-negative. It's because of the cell wall. Gram-negatives have a very thin cell wall. Gram-positives have a thick one, and they don't have an outer membrane to help stabilize things. Gram-negatives can manage to not have as much cell wall. They're fine. They were doing it with very little anyways. Whereas gram-positives rely very heavily on that cell wall. So you might not want to reach for a psyllin when you can reach for a protein inhibitor for gram-negatives. All right, we have the nucleic acid inhibitors that inhibit the synthesis of nucleic acids. These are the fluoroquinolones. They all end in floxacin. I don't know how it's supposed to really necessarily help you remember, I guess, fluorescent falcons, because fluorescent fluoroquinolones and falcons, I guess, floxacins. I don't know. Like a flock of falcons, I don't know. Whoa, which antibiotic affects nucleic acid synthesis? So we're looking, we already said, for floxacin. And the only one on this list is going to be A. All of the ones that affect nucleic acid synthesis, and that's their purpose in an antibiotic, are going to end in fluoxetine, all of them. And all of those that end in fluoxetine are fluoroquinolones. All right, so next question, why might the fluoroquinolones have negative side effects in the human bodies they're being administered to when their infections are the real targets? We're not trying to have negative side effects. Why are they having negative side effects? Because they cause the bacteria to secrete toxins, but I hope not if it's actually stopping the DNA synthesis, Fluoroquinolones stop protein synthesis by affecting ribosomes of human cells. Are they affecting the ribosomes? I don't think so. What now? I know. Okay, fluoroquinolones affect nucleic acid synthesis, which human cells also must do. Okay, this is it, right? We know that's going that direction. And then fluoroquinolones are too quickly removed from the bloodstream, not anything we talked about. This makes sense. What's happened? We don't want to go. Is that the end of it? I thought that I had another slide. Oh, I do. Antivirals. I don't know why I wouldn't go. All right, so the antivirals now. They tend to be very, we say unique to the virus. So you have to have almost always going to be treating a virus or group of viruses. Acyclovir blocks nucleic acid synthesis in HSV or herpes viruses. And Tamiflu and Relenza, they will block neuraminidase, that enzyme that helps the virus be getting into and out of the target cells. And like we said before, HIV antivirals will target these unique aspects of HIV integrase that puts the HIV's newly made DNA copy into your cells. Protease that chops up the translation product to the individual sequences to make the proteins that it needs, which is something we don't do, right? We just make the protein that we need. We don't need to chop it up afterwards to make it useful. And then we have reverse transcriptase. This is what takes the viral RNA and turns it into the DNA to be inserted in to the cell. So this is supposed to help us remember PAEC. Please, all enzymes calculate. I don't know how that must have just been left in there for something else. How does that make any sense? No, we're going to figure it out. Except apparently not, because now I'm stuck in here again. What is happening with this? Put this down. I don't know if it's me holding the pen that's making the problem or letting me write, but it's not. There it goes. Maybe not. Yeah, I did not update that one. That was my fault. One of them slipped through the cracks. The craps. Sorry. It was funnier than it should have been. All right. Which of the following, I don't have a good one for you other than just to say acyclovir and Tamiflu. And remember, acyclovir is valcyclovir, which is Valtrex. Okay, so which of the following statements is true about the mechanism of acyclovir? Does it act as a protease inhibitor? Is it an antiviral drug that inhibits replication of viral DNA? Does it bind to the viral envelope? Or is it a reverse transcriptase inhibitor? So reverse transcriptase is something we learned about with HIV, not with herpes, right? So it's not going to be this one. Then we have the envelope. No, that's just the coding, right? Then we have the protease inhibitor. Protease, again, is usually a protein for HIV, not herpes. And acyclovir is valcyclovir, which is Valtrex, which is commonly known to be used for herpes. And it does work by inhibiting nucleic acid synthesis. And that is also in the study sheet. Which of the following choices accurately describes how Tamiflu works as an antiviral? So remember, Tamiflu and Relenza, they're going to be working by inhibiting, there's on the surface of flu, the H into the N. like H1N1 or H5N1 or whatever. And those are just different designations based on their major groupings of those H's or those N's. H is going to be the hemagglutinin, which is not what this is going to be targeting. What it's targeting is the N, the neuraminidase, which is the enzyme that allows it to get into and bud off of the host cells. So that's what this is going to be targeting. That's what Tamiflu and Relenza do. So we're going to look for that choice. Does it block DNA replication of flu? No. Does it block viral hemagglutinin from engaging with target host cells? No. Neuraminidase, there it is. Just be sure if you guys are going to do it that way and be like flu, Tamiflu. Okay, we're looking for neuraminidase. Be sure you read your choices because there might be more than one that has neuraminidase. Be sure you choose the right one. And also you want to look out for things that say like something like all of the above, and it could have been more than one. Okay, so that is that for that portion of this. No, I'm not going to stand. Okay, so that is that portion. Does anybody have any questions, Jesus, about that before I move on to the study kind of effectiveness thing? Because this could be, it could go very interestingly. I have not looked over the notes I picked at all, so I didn't want to cheat, So I wanted to go ahead and make my own notes based on what you guys are actually going to see me doing and walking you through, and not have it be like me talking out my butt saying it's so easy to make a notes and then have actually looked at it ahead of time and just, been misrepresenting myself. So, right. Anybody have any questions? There's Brittany again. I'm just checking to see if we have any chat. Not yet. Okay, cool. So we'll go on to the next thing then. And get rid of this. Hey, Mama, you want to come over here? Your little dog, your little brown dog. I'll have to show you guys a picture of Ripley. I don't talk about her enough. She's just skittish. She doesn't do a lot other than be skittish. She's just a little old lady. Okay, so I picked, of course I did. I picked essentially I picked from anatomy and physiology. Okay. I have never taken anatomy or physiology, and you might think there's a lot in common. And so there can be, right? So I picked a chapter that I don't know anything about on purpose, which is the endocrine system. Okay. I know that it is involved in hormones. Guys, that's about it for me. Okay. So and I know about the chemistry behind, of course, a hormone like the steroids and things. So I have a little bit of base knowledge about that stuff. That's just going to be hard for me to find something like that. I didn't want to pick like history because I just don't have access to their notes. I would have to actually like email someone for notes about history or government or something like, I don't know, Dr. Ainsworth or something like that. And I didn't want to bother them over that stuff. So here we are with anatomy and physiology, and maybe, hey, I don't know, maybe you can relate to this situation I'm about to, you'll be a fool with. Let me pick my colors first because I need it to make sense while I'm, maybe, I don't really want this color. Let's see, I don't want that dark. There we go. Okay, let's just start with, I don't want to go, like I said, I don't want to go with something that I'm already familiar with. So let's just go start with, I guess, their action. When I saw this and I'm not going to lie, guys, I scrolled through it just to make sure there was going to be enough stuff in here that I don't know. That's the only thing I've done. But then when I saw this, I was like, uh-oh, this looks familiar. So I'm not going to lie, but believe me, I look at a lot of this and it is just, I don't know some of this stuff. I don't know how hormones work and all this. So I didn't even know there were non-steroid hormones. If it makes you feel any better, this is how I don't know. So, right, I wish I could have this be split view. And then I want another one from whatever. I can open another one. Close this one and then open it. Yeah, okay. don't want to do this right now. Now, of course, I'm like, yeah, apparently I had everything ready like in that one frame and I didn't think about this. Active notes, what did I put it? Maybe it's lectures. This is where I put it. Yes, okay. So these are Dr. Morford's classes notes, and I think that, Dr. Shearer also uses these, but I wasn't like looking for that like specifically. So this is how I'm going to look at this. I wish I had switched these. We have starting here. This is where, if this is where I was starting, you might want to start at the first slide and that's fine. But I feel like a lot of the beginning of this had stuff I already knew. And so I wanted to start somewhere I didn't know so that I could start breaking it down. Okay. So if I'm going to start off with the different kinds of hormones, and apparently there's steroid and there's non-steroid hormones, I guess. Yeah, I'm looking all over this. I probably should start earlier on. Let's do that. Okay. I'll just not focus too much on the stuff that you guys know that I already know. See how little I prepared for this. So my goal is if I was going to be taking a class that I don't know much about or it's new to me completely, things like that, then I would want to probably first things first, get an orientation of the information that I'm looking at. So this has about 79 slides. So I'd probably want to like kind of scroll through and see, okay, first they're talking about, comparisons, and they're talking about, the different kinds of hormones, maybe different abbreviations for them, the chemistries of them. Okay, get an orientation of it. That's it at first. And of course, up here at the top, they have their objectives. And I literally just pulled this off of their stuff. I have admin access on Canvas. So I just found someone's stuff and just pulled it off. I didn't warn Dr. Morford, and I didn't tell him I'm probably going to be watching one of his videos. But that's all right. I think he'll be chill with it. He's my office neighbor, so he'll be fine. But yes, so these were the goals in this class, and I haven't attended any of the classes either or anything. And if I was about to, this is how I would look at it if I needed to prepare ahead of time. This would be something where I would see if I'm having difficulty in this class, keeping up in the lecture, or if I was having difficulty, if I was online, maybe watching the videos, keeping up with that information, having to stop a whole lot, something like that. I would want to look through the slides first. The goal in this is, again, me looking through the slides first before the lecture. That's what this is. So the goal for me before having done the lecture is going just to be getting the lay of the land and familiarizing myself with the bigger overview topics. Okay, so I know in general what's happening. The goal on your look through beforehand to prepare is very general. Do not get caught up on little details unless you see something up front that you don't know. And that's what the point is of these notes. Okay, so I'm going to start off with that in mind. So here's our objectives. The objectives, I don't know how much Dr. Morford or Dr. Shearer have in their like slides, like how much they actually adhere to those objectives on that front page. That's why I took them off of my front page, because I feel like that's what my study sheet is. It tells you what you need to know. I don't have my objectives up there because I don't need you to be going through it that way. But maybe they are more stringent about it. I'm sure I would know if I was in their class, but I have no idea. So I'm not going to look at it too much now. And also, if that was important, that's probably something I would be focusing on during or after the lecture, not beforehand. So we know that this chapter is going to be dealing with the endocrine system. So I'm going to go ahead and write that down. Seems simple enough. I don't know. Just keeps you oriented or something. All right. So it works with the nervous system to maintain homeostasis. If at this point you were looking through some of these and you already didn't know what homeostasis was, which you probably do if you're in chapter 13 of physiology, it's just maintaining the stability of how everything's supposed to be working within your body to keep everything stable and functional. Homeostasis maintains your pH and, levels of ions and salts and things like this in your blood at the levels that they're supposed to be so that everything else can function. Because even if that environment isn't maintained, then the other things can't function. That would be bad. So temperature regulation and things like that. All right, so it's unique. Apparently, there's not a lot of organs that are necessarily connected, and we have some major glands. And I am going to go ahead and make note of these guys. Let me pull up my thinner. I, of course, did it on the other slide, so that's really good. Do it over here. All right, so here's what I'm going to do is in the endocrine system, make some notes of the glands. I'm going to zoom it in so I can write more easily. Pituitary. You guys are going to hear me say some stupid stuff because I don't know this stuff. The only reason I even know the pituitary gland is because a lot of horror movies they use their pituitary gland to like see into the other dimensions and stuff. That's not real, obviously. I'm talking about like From Beyond and like, I guess not From Beyond, but Reanimator and those kinds of stupid movies. Parathyroid. Adrenal. Pancreas. Oh, it was the pineal. That's what it was, the pineal gland. See, already I'm not remembering my horror movies right. Pineal. Thymus, which I do know about, by the way, because we're going to talk about that in chapter 15 and 16. And then the ovaries and testes, reproductive. So I'm just going to put reproductive. We know what ovaries and testes are. These are our glands. I have an overview of that. And I'm getting an idea of these guys. I know a little bit about what some of these guys do, like with the pancreas and insulin production, the thymus with T cell maturation, the reproductive glands with, making the gametes and creating hormones that are related to, reproductive health and things like that. Like we'll talk about that stuff, but that's about it, you guys. I don't know a whole lot. I take thyroid medication and I don't really know anything about the fact other than the fact that I have to take it. I'm just being honest here. So here it shows us the locations. I'm not going to worry too much about these things right now. It says there's endocrine and there's exocrine. So endocrine, we have internal secretions, and exocrines, we are secreting externally into, I guess. Now you see how I don't know. Now I'm having to read it, you guys. This is what you get. So endocrine glands, cells, tissues, and organs that make up the endocrine system. Ductless secrete hormones into the body fluids. Endocrine means internal secretion. Hormones act only on target cells that contain receptors for them. Okay, cool. And then exocrine glands secrete into ducts or tubes that lead to a body surface. So now we're talking about like oils and things that would go on the surface of the skin. I get this. Or like pheromones or something like that. So we're getting somewhere. And I also don't care about that. So to me, I read this. I saw endocrine glands. Okay, this secretes and it stays in your body. Exocrine glands, it secretes and it goes out to the other part of your body, outside. I might make a note of that, but I'm not even that worried about it because to me now I know that. You literally couldn't. Now I know that forever. That's a simple enough topic where I read that and you could ask me a question about it and I could definitely tell you the difference between these two after having read that. So secreting this externally, delivering our products to the surface outside of the body. So that's easy enough. Exo, outside, endo. If it was me, I wouldn't even make a note over that crap. If I tell you a term in my class and you're like, oh, okay, yes, okay, that makes sense. Sterile, I know that means it doesn't have microbes on it. Sterilization means it kills everything. Got it. I don't need to write that down. Disinfecting doesn't imply that. So automatically you should be able, even just hearing those words, you need to make a note over that. I'm not sure you even need a flashcard or write down a note or anything. To me, that is how I look at making notes. Wasting your time if you're doing more work than you need to. I'm just writing down the glands for now because I know I don't know much about them and I might need to make notes about them. So I just don't want to have to worry about it later. So we got that. Great. Here's some types of glands. I'm not worried about that right now, about pictures of them right now. That's for later. The nervous system. Okay, here we go. Comparison between nervous and endocrine. They both function in communicating, but nervous system has neurotransmitters that are going to communicate to nerves and cells nearby. And the endocrine system has hormones. So they both communicate. Maybe that's something you want to remind yourself of. That's not that surprising to me, but I'm still going to make a note of it on my little thing. Nervous and endocrine are body communication systems. I ran out of room. That's fine. And I'm not going to put a comment about neurotransmitters versus hormones because I feel like that's obvious. So we're moving on. That's going to show us the communication in nerves and endocrine systems. This is obvious to me as well. Obviously, nerves are going to be near one another and to the tissues they're communicating to and releasing the neurotransmitter communicators to the nearby tissues. Whereas I can see that, if your gland secretes something and it's transmitted to other parts of the body, then it can act on those other parts of the body, but it's not a cell that's butted up against another thing doing that transmitting. Great, cool. So, but they're communicating. I know this now. I know we have some hormones that are made by some organs that each have an organ here, hypothalamus, that is making these hormones down here. It's a ridiculous number of hormones. And this is a thing where I would be looking at this and I'm gonna be like, I'm gonna need to make flashcards for that. Which one comes from where? Prolactin release inhibiting hormone? Are you kidding me? This is ridiculous stuff. Maybe it's its own name or something. I don't know, horseshoe hormone or something. Prolactin release inhibiting hormone. I guess it describes what it does, which just seems nuts. Anyways, I guess it's better than Escherichia coli. Who am I? So whatever. So here are these hormone names that I'm not going to focus on right now, but I know I will probably need to make flashcards on later based on if your course is going to have you know those names. I'm assuming that they do need you to be familiar with those names and what, gland they're coming from. So if you wanted to, at this point, you could make a list of the hormone names. Then I'd probably say just make a card. I'd make cards that would say like somostatin, whatever that is. Sounds like sleeping. I guess it'll be somno though. I don't know what this does. Okay, so somostatin. I might have a card that literally says that on the front. And on the back, I would put hypothalamus. And I probably wouldn't worry about anything just yet. Okay. I guess these are other names for these hormones. That's nice. Oh, hey, this is dopamine. I'm learning a thing, guys. So you might make that, note that as well. Okay, so this is what it looks like when I am learning new things. Anyway, so for me too, and I have my little list, my nice little list that I'm eventually going to make into a more visually appealing situation. But for now, what I'm going to do is stop asking me that. Chapter lectures, weekly review, add new, study set. Don't care what color it is. Cool. That's fine for now. Okay, so these are in my app where I have my cards. And so my question might just be somatostatin. That's what it was. Cool. Okay, I said omatostatin. That's fine. Whatever. I don't need to be perfect. And then that would be hypothalamus, right? Hypothalamus. And that. Okay, we got it. Thanks. So I've got one little card set here. And I like this because you can make them up as you go along and build them up. So if you wanted to stop here on your pre-lecture notes so that you would at least know what we're talking about later on in the course, already have these made with the hormones and where they come from, just that basic stuff. Okay. So we could start making that. All right, back to what we were doing. It's talking about how they're released and diffused. I don't need to be reading that, and that already makes sense. Basic stuff right here, right? Steroid or steroid-like hormones and non-steroid. Okay, so this isn't a new thing. Steroid versus, I don't know anything about this. So right now I'm just getting basics down. Okay, so steroids, I know those are lipid based, but I'll go ahead and make a note about it anyways. So they're lipid based. Come from cholesterol, that all makes sense. It's going to include things like the sex hormones and adrenal cortex hormones, whereas the non-steroid guys are usually going to be amines and proteins and polypeptides and glycoproteins. A lot of this is just based off of, well, it looks like protein to me. So these are protein based, protein and polypeptide based. Helps to know chemistry, right? So those are our differences here. So non-steroid, again, amines tend to be amino acids, right? And then we have examples of each. Right now, I'm not going to worry about the examples of each, but if you wanted to, you could make another study set and literally just have a question on here of the steroid ones. I should just put this down and type on it. Versus, so those are going to be things like the sex hormones, and the adrenal, we said, adrenal cortex. You guys are going to laugh at me because I don't know what that is. Is that on your kidney? Is that the one on the top of your kidney? I don't know. Okay. Don't know where it is. All righty. This should put it in a perspective. So anyways, the non-steroid ones. Right now, I'm just trying to get it down so you guys don't have to see me typing the whole time. And this will be like I say, amino / proteins based. Okay. Oh, I just wrote that one thing and then that's not what I meant to do. This will include, what do we even say? There's all sorts of examples on here. Epinephrine, growth, oxytocin, TSH, those sorts of things. Epinephrine. I say those sorts of things like I know what they are. Growth hormone. Oxytocin. I know that has something to do with having babies. TSH. That's my extent of my knowledge. Okay, so these non-steroid, that's that. And then we'll just go ahead and put this steroid, and this will be cholesterol based. This one. Okay, whatever. You know what it means. All right, good. It gives us pictures. This is just what we like. Formulas, whatever. Hormone actions. They might have targets that they bind to, upregulation and downregulation. This is pretty similar to what we learn about with proteins and just in general, right? So if you're trying to upregulate, you might have increase in the number of receptors on the target cell in response to decrease in hormone level. And all of that can change depending on what the body's trying to do to you, but that's fine too. So I'm going to make notes about this because I'm starting to feel like an idiot about hormones. So hormones, great. What do they do and how? Hormones exert effects by, we're going to alter metabolic, deliver messages by binding receptors. They do messages. They can change the target cells. And it can affect the response affected by Number of receptors, I should say effects. It's a better way. Now you guys see what it looks like whenever you're me. So response affects receptors. So numbers. Okay, cool. So I'm learning this, steroids and thyroid hormones. Of course, they're poor water solubility. You just told me they're lipids. Transport through interstitial by specific transport methods. Of course, they would have to if they're just lipid based. We already know about membrane permeability. So you guys see how I'm reading through this and I'm already like, yeah, of course I can infer this. I already know this thing. Of course I can infer that. I know this thing. That's what we're doing right now. We can always come back during the lecture and make more notes when Dr. Morford is saying other things about this. And we can come back to this in the next week and even play some of Dr. Morford's lecture and then see how we would need to modify these notes or add to them or whatever, change them, whatever we would want to do. But for now, we just want to get the basics so that whenever we come to Dr. Morford's lecture on chapter 13, that it will make a little more sense whenever he starts talking. I don't want to know too much about the details of these, like I said, because that's something that I feel like will, I would hope that hormones have effect on how your body is working in ways like this. I'm just saying, like, yeah, I hope that they have a purpose that leads to, transcription or protein synthesis or something. But I'm not going to worry about the details of that right now. I don't need that. These guys, okay, also cannot penetrate the lipid bilayer of cell membranes. Most larger things can't because water can. That's what we learned about with osmosis and selective permeability. So these are not surprising things. Hormone is considered a first messenger, and the chemical that induces changes leading to that hormone's effect is the second messenger. So cool. So I'm getting a thing, right? So we have I'm imagining, and I might be wrong about this, but this is what I'm going to write down for now as I'm reading this. You have a hormone that gets to its receptor, whatever it is. Okay, we got that receptor, and then it's going to lead to signaling within the cell or whatever it is downstream from it. That's going to lead to more signaling. I'm just going to write that. And this will be our lipid bilayer. I'll put inside of cell, and this will be outside cell. Okay, cool. So this is what I'm getting from that whole statement. Tell me if I'm wrong. This is the first, first messenger. I hold my pencil very tight, and sometimes it just pops up with the thing. First messenger. And then this is the second, whatever comes after the receptor, right? Everything that is going to be the cascade downstream from that interaction with that receptor by that hormone. Okay, cool. So this is how non-steroid hormones are going to work. What did I miss on the steroid? So these boys can diffuse the lipid bilayer. So they can work directly. Okay, I'm learning a thing. Okay, so so this is, let me get a different color. Let's do black. Sure. So this top one here, and first let me move it because I need a little more space. I want to, for those of you who are probably bored, but I mean, if this isn't like telling you guys kind of how to be thinking through this, I don't know what to tell you. Here's my color. Oh, because I have to choose the thing first, of course. Okay, so this was specifically the non. They have to act through a receptor. If you're steroid based, then what you're going to do is your steroid hormone. And here's the inside and the outside. Outside, inside. You're just going to cross this membrane. You guys probably honestly know more about this than I do. This is a thing, okay? So these guys are going to cross the membrane, and they are then going to be able to affect genes directly. So I'm going to just put act on genes. I'm going to circle that. So okay, so this is what I'm getting from that. Hopefully I'm right. Yeah. Here we go. So it's showing me a picture of what is looking like transcription and translation. And we have a hormone receptor complex, whatever, that'll be acting within the nucleus here. So that's what's happening. Just so you are seeing what I'm talking about at the same time, right? And then it affects the DNA expression. So that's great. So we're kind of getting this. I don't need to do much on the details right now. And so that makes sense. Non-steroid guys, they got to act on this receptor and trigger something downstream to affect the cell. Okay. So people do abuse hormones to improve athletic performance, growth hormone, erythropoietin to make more red blood cells for training. Okay. So I don't care about that right now. Prostaglandins, paracrine substances. Sorry, I know nothing about this, so I'm not like reading about this. Rapidly inactive attributes regulate cellular responses to hormones. Okay, well, I'm going to make a note. Actually, I'm going to make a study set thing because I don't know anything about that. Prostaglandins. So I know for sure that's something that I'm going to need to be whatever. Needing to look at. So that's all I'm going to put is regulate responses to hormones right now. Yeah, that's all I'm going to do. I know I just typed that wrong, didn't I? This also implies to me that they themselves are not actually directly hormones, right? Because we have like hormones, we have responding to hormones and stuff. So that's not a hormone, but it helps in the hormone response. Cool. Learned a thing. Next part. All right. Let's see where we are on this. How are we going to control these? Negative feedback. I'm not too worried about this. So let's start with negative feedback. I'm going to start another sheet. All right, so rising level of a hormone leads to decrease in hormone secretion. Hormone is used up, inhibition stops, and secretion begins again. This will keep needing it. So it's basically controlling how much hormone is being made based on it being used. No. I say, oh no, I just don't like it when people come to my house. And that's saw somebody. Okay. Cool. So three methods of negative feedback control of hormone secretion. We have the hypothalamus. So I'm gonna make it like this. Hypothalamus. Oh my gosh, this is a lot. So this is something where maybe I'm not going to go into the detail on it. So I'm going to say maybe just say hypothalamus. Okay, so it controls the release of anterior pituitary hormones. Then pituitary hormones secrete hormones that control activity of other glands. So I'm going to say cascade effects of the hormones relating to the anterior pituitary or something like that. I'm going to put anterior / pituitary. And etc. Because I don't need to know too much detail right now for my prep. Okay, nervous system control. Nervous system directly stimulates some glands to secrete their hormones. This makes sense to me, so I'm just going to write just that. That's just what the nervous system does, right? Right, and then we have changes in composition of the internal environment, changing levels of a specific substance in the blood stimulates or inhibits secretion of certain hormones. Okay, so I'm going to put internal environment. And then I'm going to just clarify by only just saying non-hormone molecules or substances. I like that better. Affect hormone levels, right? Okay, and then below this, we have positive feedback. And it doesn't seem like it says very much, so we don't need very much space. And it says mechanism in which rising level of hormone leads to an increase in secretion. It's not used very often, it doesn't look like. So increased hormone leads to increased secretion. I'm not going to worry too much about the details on that right now. So cool. So here we have some pictures of some of that working out. Negative feedback. I'm not going to get into the details of it right now. We'll worry about the details whenever Dr. Morford explains it to me. So now we're into the pituitary gland. So it's in the base of the brain, attached to hypothalamus. I don't know any of this, by the way. I mean, I knew it was in the brain, but I don't know where. Base of brain. Attached kind of to the hypothalamus. I'm just going to put that abbreviation. Attached by the pituitary stalk. Oh, good. This is where I hate you guys for taking anatomy because I don't want to learn these words. Okay. Already, I don't know what that is. What word that is? I've never heard this in my entire life. And I am definitely not going to be knowing what these are. Sella turkey. I don't know what these are. Okay, so we're going to do this. Pituitary stalk. I'm already behind in my class, guys. Look at that. And then this is the infundibulum. Do I sound like an expert yet? These other things right up here lies at the base of the brain in the sella turcica of sphenoid bone. Don't know what that is. So since I really literally don't know what that is, and I might want to know what it is, especially if I was in the class for real, right? Then what I want to do is I'm going to get like, let's do this color. That sounds good. Sounds pretty. So that I see this one because I know I do not know this. And it seems like something I should have just known. Sella, whatever. Don't know what these are. Okay. So we've got two parts of our hypothalamus, or sorry, two parts of our pituitary. Oh no, I changed the blue that I was using. No, we don't need gray. Maybe. Um, pituitary gland. We've got two parts. The anterior and the posterior. This is fine. I know what these words mean, so I'm not going to worry about that. Adeno hypophysis. Yeah, you guys are killing me. Good. Stomach is growling. Okay, anterior. Adenohypophysis. I'm probably saying this like a fool. And posterior. I don't know how important that is right now, but I want to write it down because I have never heard this. All right, so we have the small intermediate lobe called the pars intermedia, develops in the fetus and it's melanocyte stimulating hormone, which starts melanin production. And then I'm gonna just put this, okay. I'm just gonna put melanin production for now. I'm not gonna get too much into these details because I'm gonna get lost in it. Okay, the hypothalamus is gonna control it. There's a lot of listing of what sorts of hormones in order are going to be doing what. This sounds like something that I'm going to need to write a chart up about, and that's okay. I'm not going to worry about it too much. Then that was something that we'll come back to probably later on. This is what I'm talking about. Yes. Okay. So we got that. So, releasing peripheral endocrine gland and negative feedback. So I'm not going to get into too much of that. Let's see. This is clearly going to be more helpful than the notes that I'm writing. So I'm going to take a picture of it and put it here. Did it work? No, it didn't. so that I can look at that and have that there. Please just let me make this smaller. There we go. And I can always make it bigger. Like I put it on here small. I can always make it bigger later if I need to and make it larger. Where are we? About halfway. That's not so bad. About like halfway through the hour. So I'm probably going to make it through this whole thing in an hour. Again. So releasing hormones of the hypothalamus. So these were our releasing hormones. And our term for that was they act on specific hormone secreting cells in the anterior pituitary gland. Okay, really. So I'm going to put hypothalamus. I'm going to put releasing hormones. I'm going to put A for anterior, A pituitary. I don't know what that means. Okay, so this is really, my handwriting's getting better and better as we go. And then we have talking about these guys. Here's some names of them. And then here's what more details about them. So I'm going to just make a list for now. Growth hormone, prolactin, TSH, adrenocorticotropic hormone. Great. Great. That's great. And then follicle stimulating. I know what that is. And luteinizing. That's how I say it. Probably not pronounced correctly. Who cares? FSH and LH. Okay, cool. So I kind of know what some of these are. At least I've heard of them before. Great. And this is just showing how they're going to work together and act on things. This is kind of a useful one. I might take another picture and put this one. How they're going to act on organs in general. So let's put it. Oh, I made a really big one on this one. I didn't mean to do that. First. Sorry, I didn't mean to make this so big. I'll keep up with me. Thing, are you serious? Why is it not letting me? I don't know. I don't know if I can do it because it's not letting me draw over there. Yeah, it's not letting me go over there for some reason. It's fine. Okay. Great. So here's the function of each of these. I have this picture and it gives me like the gist of where they're going to operate. So for me right now, as far as like, okay, it tells me about prolactin, promoting milk. And so I can see that here on my little image that I have. Let me get, I think let's do that here. That whenever I look here at prolactin, it's telling me that already. So I'm not going to make any notes about this, just so you know why I'm not me being lazy. What the gist of it is already there for me. So thyroid hormone secretion, We already were told that it's made by the TRH is secreted by the thalamus that causes the anterior pituitary to secrete TSH. And then we have this sort of stuff happening. Again, the picture is more helpful than anything that I'm going to be writing down. And because I kind of get the idea of how these are going to work together and keep signaling and triggering one another, I feel like this is probably sufficient for now. If you guys see images like this on your own and it doesn't make sense after you glance through literature, then don't just put it in your notes. Take a minute to try to understand what it's talking about, because I'm doing that here. It's just I'm not talking about it much. So I am actually perusing this. Okay, this is stuff talking about how the thyroid is going to get stimulated by a hypothalamus and the anterior pituitary. So this is showing me that little pathway here. So now this is talking about some differentiation of what we would see in people with growth hormone problems. And I'm not going to get into that right now. I think that's more specific than we need for our preface leading into the course. Posterior pituitary hormones. Okay, hormones made posterior lobe. They are transported to the posterior pituitary gland via the pituitary stalk. Again, we are there. I get that. And then posterior pituitary hormones are going to be antidiuretic and oxytocin. So still regulated by the hypothalamus, yes. Okay, cool. So we can just add on to that. Move this again. I guess that's coming with us. Move this down because I want more space. Where is this? No, I changed my blue again, you guys. I don't even really like this pink. Okay, so we have the posterior pituitary hormones. It just does this thickness thing because I shrunk the other one. And so this is going to be antidiuretic hormone and oxytocin. Not going to worry too much about details about what's going on with these guys right now. I'm just going to put vasopressin because I feel like that's good to know. Right, so now in my note taking, what I'm going to do is keep going, making sure all of this is making sense to me so far. And then now we're talking about the thyroid gland. Great. So we have the parts of this one. Where are we at on this thing? Two lobes connected by an isthmus. And I actually know what this means, so I'm not going to make a note about this one. So thyroid gland, the two lobes, three hormones. Let's go down here because we're already under like an area that makes sense with all of this. So thyroid. Two lobes, apparently, lobes. And then we're going to have our three hormones. T4, T3. I don't like that. And calcitonin. So this one's also known as thyroxine. Thyrine. That's a weird word. Great. So we've got secretory units called follicles. You wanted to see that really big, right? That specifically. Okay, there's our gland. I'm good with this. Here's some pictures of some cells. Here's some talking about the action of these thyroid hormones. I don't know how much I want to get into that, but this might be a good one for a study set. Is it going to talk about it? Not really. Okay. So I am going to do that then. I'm going to go to my study set and I'm going to start adding just those in there. So T4. And then on the bottom one, I'm going to call it thyroxine. I actually take Synthroid, which is levothyroxine. Then increases rate of energy release. I'm just put energy from carbs. I'm going to put protein synth for now. Growth and nervous system. All right, our next one is, let's see, T3. This is also known as this word, triiodothyronine. And then this is thyroxine, but more potent. Okay. And then I have calcitonin. This is blood calcium. Relating to bones. That's all I'm going to put for now. Oh, come on. Oh, come on, please. Increase. This is fine. From kidneys. Okay. Great. Good. I'm happy with that. All right, now it's going to talk about me some disorders. I don't really want to get into that right now because I'm just trying to learn about the basics, and then we can learn about that during the talk. Right. So parathyroid, posterior, usually 4 parathyroid glands. Okay, so this is posterior of thyroid. And then it only secretes PTH. So we know what parathyroid hormone is. So PTH, I don't know what it does, but I've heard of it anyways. So it increases blood level of calcium, decreases phosphate, bones, kidneys, intestines, and vitamin D. So increase blood calcium, decrease PO4. We have some steps in vitamin D metabolism. Good. I'm happy with this. If I care about it, then here I'm going to select so I can look at this process while Dr. Morford lectures. God, I made it so big again, you guys. Wish I would just pay attention to what I'm doing. I just don't. Save me the time. Can't. Great. Okay, good. My parathyroid BS. And then we've got more pictures of how it's going to be acting on things. I like this one probably even more than I like the other one. So that's great. Just put that one as well. You guys get to see pictures of my dogs and stuff while I pull out pictures of things that I